Hello, everyone. Basically, we're going to go over a topic called the scientific method. So, I will have something called a lab introduction for almost every lab that we do. Okay, in most cases, you're going to basically get a piece of paper with some writing on it, and then you'll have to fill in the details as I write them on the piece of paper, or if we're in person, as I write them on the board. So the first lab that we do is going to be called the scientific method. Okay, and it's going to discuss steps that are taken within the scientific method during any type of research experiment, whether it's simple type of research experiment or whether it's a complex type of research experiment. So I'm going to use Cheerios as an example for an experiment. So the reason I got this idea is because a long time ago, years ago, okay, I saw commercials that claim that consuming Cheerios reduced cholesterol levels. So basically, I came up with this idea of a Cheerios experiment to demonstrate the steps of the scientific method. So, what we're going to do is we're going to talk about 5 steps within the scientific method. Okay, listed right here 1, 2, 3, 4, 5. Okay, and then we're going to go ahead and we're going to add some details into each step. So, um, the first step of a scientific experiment is always going to be an observation. So, let's go ahead and write that in. So observation. So you're going to go ahead and write observation into the first step of the scientific method. Okay, and the observation can be a very simple observation, or it can be, a detailed scientific observation. So it could be something as simple as, hey, I've been eating Cheerios a lot and I noticed that my cholesterol is lower. Or it could be something really detailed and complex, such as, hey, I'm a biochemist and I know the chemical composition of a Cheerio and how that interacts with the components of the blood and how that interacts with the cholesterol that's in the blood. So you can have different types of observations. But the first step is usually going to be some observation that gives a person or a scientist an idea for an experiment. So now we're going to move on to understanding step 2 of the scientific method. Okay, so step 2, in order to complete step 2 of the scientific method, we are going to pretend that you are the head scientist at the Cheerios company. And the marketing department comes to you and says, hello, Mr. or Mrs. Head Scientist of Cheerios. Can you please plan and perform an experiment that shows that eating or consuming Cheerios will lower a person's blood cholesterol level? So you go ahead and you get to work on planning, okay, this experiment. So you're going to formulate something called a hypothesis. Okay, a hypothesis has been known to be called an educated guess, but I like to call a hypothesis a testable statement. Testable statement, a statement that we can test. So I'm going to come up with a couple of hypotheses for our Cheerios experiment. Okay, and in lab, you are going to come up with some hypotheses for different experiments. Okay, so a hypothesis can usually be written in an if-then statement, as an if-then statement. So for example, if Cheerios are consumed, if Cheerios are consumed, then cholesterol levels will decrease. So notice that this statement has the 2 words if and then. Okay, so that is a statement which we can definitely perform an experiment to test. Okay, now I want to make sure that you understand that a hypothesis does not have to have the words if and then, okay, within the statement in order to be a hypothesis. So, for example, Cheerios consumption will lower cholesterol levels. Cheerios consumption will lower cholesterol levels. Okay, that definitely is another statement that is testable, but I did not use the words if and then. So I just basically do not want you to get hung up on using the words if and then, okay, in your hypothesis statements. Okay, so the third part, okay, of the scientific method is to plan and perform an experiment. Plan and perform an experiment. Well, for the Cheerios experiment, you're going to need some people. So let's say the Cheerios company rounds up 1,000 people for you to perform your Cheerios experiment test on. So, what is going to be the first thing that you do with that 1,000 people? Okay, basically, one of the first things that happens when we're starting to plan an experiment is we're going to take the individuals and divide them in half. Okay, so we're going to have two groups of people. So divide everyone in half and form two groups. The first group is going to be called the experimental group and leave a little space right here. And the second group is going to be called the control group. So, which group do you think is going to consume or eat the Cheerios? Okay, that's right. The experimental group is going to consume Cheerios. So then why have the control group at all if they're not going to eat the Cheerios? Why bother having a control group? Basically, people like to compare. Okay, comparison, when you compare something that is kind of really strong and persuasive data. So the control group allows for a standard of comparison. Allows for a standard of comparison. So now you have 1,000 people, 500 people are in the experimental group, 500 people are going to be in the control group. Okay, so the only difference that you really want to have between the group is that the experimental group is consuming Cheerios and the control group is not consuming Cheerios. Okay, so the difference between the two groups is called the independent variable. Independent variable. So the control group is going to contain the variable, which we call the independent variable. So the independent variable is basically the difference between the two groups. So that's the difference between the two groups. We want to try to keep everything else the same as much as possible. Okay, so we call all of the variables that we want to keep the same between the two groups controlled variables. So in our Cheerios experimental group, let's give some examples of some controlled variables. Okay. So basically, we may want everyone to have the same cholesterol level at the beginning of this experiment. Okay. So same cholesterol level at the beginning. may want to have everyone be the same age. We may want to have everyone drink the same amount of water. We may want to have people that have the same or at least similar medical histories. We may want to have either all male or all female. Okay, so basically, we can go on and on about the controlled variables, especially when we're talking about people. Okay, because there are so many differences between people, and everybody is really, really unique. This is why it makes it really hard to test, okay, on people. But we're going to try to do the best we can to control all the variables between people. Okay, so the goal of that is so that we can prove with even more significance that consuming Cheerios did in fact lower cholesterol. So we're going to talk about one more variable. It's going to be called the dependent variable. Dependent variable. Okay, so the dependent variable is basically going to be what you are testing for. In this case, we're going to be testing cholesterol levels at the end of the experiment. So in our example, it's going to be the cholesterol levels at the end. Cholesterol levels at the end of the experiment. Okay, so basically the dependent variable is going to be the variable that you have no control over that. Okay, so you do not have any control over the dependent variable. You cannot control the dependent variable. So you're not going to be able to control the cholesterol levels of the people in this experiment at the end of the experiment. Okay, you're basically seeing, how the consumption of Cheerios affects the cholesterol levels. So you want to make sure that you understand the independent variable. And a good way to think about that is the independent variable is going to be the variable in the experimental group. The dependent variable, okay, which is going to be the variable that you're ultimately testing for. We're recording cholesterol levels at the end of the experiment, and we have no control of those cholesterol levels. So that's going to be what we call the dependent variable. You also want to keep in mind the controlled variables that you're trying to keep consistent between the experimental and control groups. Okay, so step 4 is going to be, let me move this up a little bit. Okay, so step 4 in the scientific method is going to be the results. So let me put a little line here, sorry. Okay, so results. And sometimes the results are also called the data. Okay, so in this case, it's pretty simple. Okay, we are basically going to record numerical data since blood cholesterol levels are basically numbers. Okay, so numerical data is going to be the type of data that we frequently see, okay, in research. Numerical data, okay, for example, blood cholesterol levels. Okay, we call numerical data quantitative data. Okay, so quantitative. So in our Cheerios experiment, we're basically collecting quantitative data, and that's going to be the result of our experiment. Okay, you want to keep in mind that not all data at the end of an experiment is quantitative. For example, if we're looking for the color of something, maybe we're doing an experiment for flowers and we're trying to look for the color of the flowers. Okay, maybe we're trying to do an experiment where we're testing fragrances. Okay, maybe we're doing a taste test. Okay, so basically that is going to be what we call qualitative data. So if you're looking for color or fragrance, which is basically smell, or taste, etc. Basically, anything that uses your special senses. Okay, that's going to be called qualitative data since it is not numbers. Okay, so but in our experiment, our Cheerios experiment, we are collecting quantitative data, and that's going to be our results. So the last step of the scientific method is going to be forming the conclusion. Forming the conclusion. And within the conclusion, you're going to either um, accept, which we can also call support, or reject the original hypothesis. Okay, if we reject the hypothesis, let's say in this example, it would be cholesterol levels were not significantly lowered by eating Cheerios. In that case, we would be rejecting the original hypothesis that we had. So, in the case that you reject the hypothesis, what scientists normally do is they normally kind of like come up with just an idea for another hypothesis. Which ultimately could possibly lead to another experiment. Okay. So if the cholesterol levels were significantly lowered, okay, in the experimental group, which basically consumed Cheerios, then we would either accept or support our original hypothesis. Okay, so I want you to keep in mind that these are very basic steps in the scientific method, and in your lab book they do expound a little bit and include some more steps within the scientific method. And basically, you're going to read a bunch of different types of experiments and identify components in the scientific method.